Protein synthesis is the process through which DNA can result in the production of a protein. RNA has a significant function in protein synthesis. These are the two key stages. One, transcription, is generating single-stranded mRNA from DNA and happens in the nucleus of eukaryotic cells. This takes place in 3 stages of initiation, elongation, and termination. 2, translation, is a procedure through which mRNA's codons are converted into a series of amino acids. By complementary base pairing, a component of the tRNA, known as an anticodon, recognizes and attaches to the codon on the mRNA. Messenger RNA is a molecule that is produced in the nucleus of DNA and travels into the cytoplasm where it interacts with a ribosome. The translation of an mRNA into a polypeptide occurs at a ribosome. The 3 stages of transcription are: 1, initiation, is the section at the start of a gene known as a promoter initiates transcription. 2, elongation, when mRNA polymerase unravels the DNA fragment, transcription begins. 3, termination, a stop signal is encoded by one of the 3 triplets when the polymerase reaches the end of the gene, signaling the enzyme to end transcription and release the mRNA transcript. The cytoplasm translation takes place. mRNA needs to undergo several modifications after exiting the nucleus before being translated. Iotrons are regions of the mRNA that do not code for amino acids and are thus eliminated. One end of the mRNA receives a poly-A tail made of multiple adenine bases, and the other end receives a guanosine triphosphate cap. mRNA is ready for translation when all changes have been made. The crucial task is carried out by the nRNA, a mechanism that produces proteins. RNA creates the essential structure of the ribosome. From the DNA, messenger RNA transports genetic material to the ribosome. Ribosomes link together to transfer RNA-bound amino acids using information from messenger RNA. The mRNA transcript leaves the nucleus once it's been prepared and moves to the cytoplasm. A ribosome is a microscopic, non-membrane bound organelle comprising of RNA and proteins. Ribosomes serve as the site of translation during the formation of proteins. The genetic code contained within the mRNA is processed and translated by the ribosome. This is where a protein's amino acids sequence is created. The small subunit and the big subunit make up a ribosome. The big subunit is in charge of catalyzing the creation of peptide bonds between amino acids, whereas the small subunit is in charge of decoding the mRNA. The individual components of the protein synthesis are called amylines. During the process of making proteins, transcription occurs in the cell's nucleus. This turns mRNA into DNA. The ribosome subsequently receives the mRNA. The function of transfer RNA is to convert the information contained in the nucleotide sequence of the mRNA into a particular amino acid sequence. A region on the mRNA, known as a codon, that the anticodon specifically recognizes, and mRNA molecule is created by transcription within the cell's nucleus and is changed. With the assistance of a ribosome and tRNA molecules, the transcription is translated into a protein. tRNA is a form of RNA that transports the essential amino acids to the ribosome and joins each amino acid to the previous one, constructing a polypeptide chain. As a result, tRNA transfers amino acids to the cytoplasm into a polypeptide. The final product of translation is a chain of amino acids that were introduced in certain sequences according to the mRNA's coding. DNA is in charge of directing the protein building process, but mRNA, tRNA, and rRNA are essential. A polypeptide is a name for protein. A protein's 3-dimensional structure is determined by where each amino acid is situated on the lengthy chain of amino acids that make up a protein. The sequence of amino acids in every protein is distinct. The polypeptide backbone is the recurring patterns of atoms that run down the center of a polypeptide chain. Depending on the structure and function of a protein, folding and modification may take place. My 2D model of a flow diagram effectively explains the process of protein synthesis in a logical, coherent, and simple way. It has allowed me to have enhanced clarity on the process. Advantages are it provides knowledgeable reasoning supported by evidence from trustworthy academic resources. Disadvantages are that the physical limitations are left out, and it was time-consuming to create. The sources I selected were reliable, since they offered a well-supported argument which were based on evidence. Some were peer-reviewed and published by universities. These sources are valid because they can be trusted, as they draw valid conclusions which have been confirmed against other resources.